Thanks for coming along, everybody. We are the Mur Ligase team, and it's the 11th of October, 2022. And I will just share the right thing, which is here. Whoops. Okay. So hopefully people saw the issue I shared, and it outlines, lots and lots of things which are going on. To my mind, of all the things we're doing at the moment, which are listed here, the things I want to talk about most would be latest results from you guys, Laura and Adrian, and also just comments on the latest structures that came through, and just to prefigure that perhaps, given all the new data we've got recently, we might want to have another dedicated session on crystallography. So soak in number 2, but we could talk about the new structures and what we learned from those and what we need next, if anything, from the SSC CRD guys. So if you're happy to, the Warwick team, to take over and talk about what you've been doing recently, that will be, I'm sure, of interest to everybody.
I'm on mute. Yeah. Yeah. Should I share?
If you've got something, that would be really great. Yeah, you've seen, I think people have been inundating you with compounds recently. So
can you see the?
Yeah, all good.
Should I start with?
And you want some videos?
So I've been doing some SPR the past 2 weeks, I think. No, last week. Yeah, week and a half. So I've got some results for SPR with E. coli Mur D on the APO confirmation with the competition compounds for this first round. So I've got some KD values, but take them with a pinch of salt, right? Like I would say this is 100 micromolar, 100. I will say these are very similar because the fitting is not great because there's more data points to add. But we got one compound, this compound 4, that is at 10 micromolar binding, as you can see here.
Wow.
That's the curve for it. Which is looking more promising than the rest of the compounds in this screen, and then the rest they look more like this. Like I can't see the saturation mainly.
Okay.
Yeah, there are some that don't have. I can only see non-specific binding, so that doesn't mean there is no binding. It's just that the only thing I can see is the non-specific binding, and there is some that I couldn't detect any binding.
Right.
Yeah.
Sorry to jump in. So just 2 questions immediately. One is, when are you happy to sort of conclude this? Are you wanting to do more repeats, or are you basically done with the repeats and you're happy with that sort of 10 micromolar value?
It's done now.
It's done.
It's done. This is the second time I've did 2. I've got the same result twice.
All right. Awesome.
Yeah, and I don't want to kill the sensor. I want to redo the other things.
Yeah, no, that's great. So the second thing was just, how do you determine that it's non-specific binding? Sorry.
So the non-specific bindings, they look like a straight line, basically. Now, in this binding, right, you cannot determine which one is a specific and which one is not a specific. It's just how the curves are looking. It might be a mixture of both, but we are never sure about that. That will be with any binding experiment, basically.
Right.
You never know if you're binding to 1, 2 sites or 3 in these kind of things, especially with low binders that you cannot discriminate within different binding sites, if there are any. So sometimes you can see tight binding and then the non-specific binding, and you can separate both of them. But in other occasions, that's not the case. Yeah.
Right.
Yeah. So these are some results. I can share them on the GitHub after the meeting.
So it looks like there's, one sort of 10 and a few are about 100 kind of thing.
Yeah, quite a few about 100. There are some that are more than 500 micromolar. That's the top concentration.
Yeah.
So yeah, obviously I don't trust the values that it's giving me, so it's higher than 500.
Yeah.
Yeah, and the rest around 100, and then we got the 10 micromolar.
Okay. Really good, thank you.
Just a reminder, this is with the APO without any substrates or ATP bound or anything.
Yeah. Sure.
Yep.
Sure. And what's the possibility of having compound 4 put into a soak or something of that kind, just to see if we can see where it's binding?
So soaking experiments will be done next week.
Oh, awesome.
We are hoping to send crystals at the end of next week, but we are coordinating with another team, so let's see how that goes.
Okay.
So hopefully I'll have soaks I will soak everything because I'm preparing crystal plates for many compounds, so I can soak them all.
Great, thank you.
Yeah.
Good.
Adrian will update on the rest of these. And I have also started the enamine library screen on the SPR. So I mobilized Pseudomonas Mur C, Mur D, and Mur E on the sensor, and I screen the whole library, and I've got some hits. This is the map, the hit map, basically. So we've got some potential binders that could be non-specific binders more than specific, because these are really high values. But I'm currently running the dose response for the best 32 compounds that could be binding to at least 2 Mur ligases. So I'm doing that as we speak. So I'll have the results tomorrow, because the run is like 32 hours. And I'll have that tomorrow, and I'll share the data as soon as I'm done analyzing the data, basically.
Amazing. So these are the compounds from the Warwick enamine collection, right? Not the atomized ones.
Yeah.
And
Becca did, right?
Right. Yeah. And I guess what we're hoping for is a correlation here between the SPR hits.
Exactly. So far, I haven't seen the correlation.
Really? So you're getting SPR hits on things that don't enzymatically inhibit?
Well, not at the level, so I cannot find the final hits from that we are following up. The best hits on the assay, I can't find them on the SPR as hits. But we're going to look after I do the experiments on the dose response, and I make sure which ones are non-specific and which ones are specific. We will look again into the hits, and we will look again into the initial screening hits and see if there is partial inhibition for any of the compounds that are giving a hit on the SPR. And then obviously share that.
Yeah.
Yeah, so that's on process.
Okay, great.
And now, Adrian, if you want,
Okay.
I'll put this closer to you.
Done. Okay, so we've received the competition compounds a couple of weeks ago, and we were under the understanding it was fairly urgent to get them processed as quickly as we could. So that's what we've concentrated on. So what I've done, I've screened all 18 compounds against Pseudomonas aeruginosa Mur D in the standard Amplex red assay that we use for screening. And I repeated the screen against the coupling system that we use, which is the sequence of enzymes that generates the fluorescent signal that we follow. Now, the initial screen against Mur D, Pseudomonas Mur D, was quite encouraging. There were a large number of compounds which gave significant inhibition. And compound 4, for example, actually gave startlingly complete inhibition under the conditions that we used. When we looked at the impact, however, of the compounds on the assay itself, as opposed to just Mur D in isolation, it became clear that most of them actually targeted the coupling enzymes as well. So at the concentrations we're working with, and these are one shot at 1 millimolar, there's no real specificity. So what that basically means is that I can't hand on heart say that the data in the top panel against Pseudomonas Mur D is an actual reflection of the impact upon the enzyme. So what I basically have to do with this is back off the concentration of compound, because if, as Laura says, we are getting 9 micromolar KDs, I should be able to do that and still see an impact. To see whether we can actually have a window that allows us to operate in between the sensitivity of Mur D and the sensitivity of the coupling system. Failing that, then we need to go to an alternative assay, which we do have, which we use for ADP release, where we use pyruvate kinase and lactate dehydrogenase. So, talking of which, There's an atom-wise library update. So this concerns the compounds that did inhibit the amplex red-based coupling system, the ones that we, in the initial phase, had to discard because of that. These 15 compounds were re-screened with an assay that follows ADP, pyruvate kinase lactate dehydrogenase, and in actual fact, 14 of these at 1 millimolar still significantly inhibited the coupling system. One compound, however, AW16, which has the title Z917-17000, was actually a weak inhibitor against C and D and E. coli Mur E. So we can add that to the collection of atom-wise compounds that have an impact upon multiple ligases. So otherwise, so in the background, a number of things have been happening. For example, the synthesis of substrates, the establishment of a Mur assay, which we've done. And we've now, I've got to process the data, but we have dose response curves for Pseudomonas aeruginosa Mur versus our control inhibitor, ADPCP. We need to characterize the enzyme kinetically before it goes into the enamine screen. And finally, I mentioned this before, that we have the idea that molecules like uridine tetraphosphoadenosine, which have a uracil and an adenine nicely spaced between them, might actually impact a Mur ligase at both the ATP and UDP substrate binding site. It turns out that UP4A is, of all the Mur ligases we've tested, a specific inhibitor for Mur E. It also turns out that AP4A is a submillimolar inhibitor also of Mur E. And we're doing dose response curves to see if there's anything there that's worth pursuing, because it might be quite nice to develop a molecule which was able to actually target both the uridine and adenine binding sites from a Mur ligase. And that's where we are.
Great. That's wonderful. Very interesting. Thank you. Anyone have any questions about what was just presented from either Adrian or Laura? Yeah, I guess the competition compounds are odd, right? So I remember that from the atom-wise library, yeah, I mean, that there, as you said, there were 15 compounds that appear to mess up the coupling enzymes. And it's the same issue you're talking about with the competition compounds, right? That they're doing the same thing?
Yeah, it appears that we're seeing the same sort of issue.
Right.
We didn't really see it anywhere near the same extent with the enamine library when we screened it against Allegalactii. But we did see a significant, well, 15% of the molecules in the atomized library appear to be non-specific. And pretty much all the molecules we've received in the competition appear to also be non-specific at the concentration we're working at. And I think I need to stress that point. If we drop, because we're using high concentrations, because we've been habitually trying to detect fragment binding, although we have some intelligence from Laura that we do have molecules in the competition which bind far more tightly than would necessitate use of 1 millimolar as a concentration. I think the next thing we will do is back off the concentrations of what we're using, just to see whether, as I say, we can open up a window that we can actually exploit.
I guess I'm just interested in why the competition compounds are all hitting. Is it just concentration thing? So we're using them at a higher concentration than the atomized compounds?
No, not especially.
Is there any reason then you think why the competition ones are sort of 100% hitting the coupling enzymes?
To be honest, I don't know off the top of my head. I don't have. We would have to look. I mean, we could actually start to investigate further and look at the impact on individual other enzymes. I mean, the other possibility is that we try and steer away from doing coupled assays in the way we've been doing for these compounds, and maybe look at some of the chemical methods for phosphate release. The reason we haven't done that is those methods require use of a fairly high concentration of acid to get color development, and I have a problem with that because the UDP Murnac peptides break down significantly and also will liberate phosphate under those circumstances. So that's a can of worms that I wanted to try and avoid opening if I could avoid doing so.
Right. So the easiest thing to do is back off the concentration, see if that helps.
Yeah.
Okay. Yeah, I just wondered if there was something about the samples or the the DMSO, or I don't know what it was that we have the compounds in, but I think they're all pretty different. So
Yeah. Well, I mean, if there is some common solvent trace left over, I don't know. I mean, it's
I doubt it. No, I doubt it.
I.
I mean, yeah. I mean, some of them are from enamine, right? I think the compound 4 is from enamine, actually, if it's got a Z code.
Yeah, that's true.
Okay, great. Thank you. Very interesting. We will take a look at the structures for interest, and we'll give an interim report, but we'll make it clear that it's a work in progress.
Yeah.
I mean, it's very nice that there is apparently something there from SPR, which really helps. We don't know where it's binding or what it's doing, but that's encouraging. Yeah.
I could also see binding with Pseudomonas murdi as well, with compound 4.
Really? Okay. Yeah.
That is, Pseudomonas murdi, well, it's expected, but
Right. Yeah, that's great.
Yep.
Okay, great. Any other queries about the data? That's pretty nice. Okay, great. Well, that's I guess that's all of these issues here. I suppose the soaking you're doing, Laura, is with these obviously, but also other compounds. I guess we had the elaborated fragments that Ed sent as well. Are those also included in soaking, or I guess we need to be careful that we're tracking all of the different things that we sent you over the last few weeks and months.
yes, if I have enough crystals, obviously I'll try everything I can. I want to try again the hits that we're getting with the essays.
Yeah.
These competition compounds, and then anything I can.
Right.
Anything that it hasn't been tested, obviously.
Yeah, I mean, I think on the fragment elaboration stuff that Ed sent, were the compounds he'd made and Cato had made. We don't yet have data on those, I think, from anything as far as I remember.
I don't think so.
Fine, though, because that was the next phase of using the fragments that we had, right? So we had some hints that they were multi-targeting, and we'd made a bunch of semi-random derivatives based on what we thought was best. Okay, so those are awaiting evaluation in something. Okay. I mean, I guess, we've got excited about the Warwick enamine collection, the atomized compounds. We've got to remember the fragment side of things too. Okay, great. So this is a work in progress with the atomized compounds, so we don't need to cover other things about going back to atomized just yet until we've completed the data. All right. Just on the shipping of compounds, I think Yuhang has finished and now shipped this key amine derivative of the original AZ hit. Yuhang, did you want to say anything about this, or have you already posted the something below? Because essentially you finished the synthesis of this compound, which has been a real pain to get hold of, and you've shipped this compound now, right?
Yes, we got it already.
Oh, really? Wow, that's quick. Okay.
Yes.
Great. Okay. I mean, obviously, this is Yuhang's target for a while now, and it's been really quite challenging to make. But I'm pretty sure that we've got it. It looks on the NMR like it's the 4-mate salt here. So there's a NH3 plus and then a 4-mate anion. That just needs to be factored in terms of the molecular weight, but it doesn't make that much of a difference.
How stable is it?
Well, as the salt, we think it's fine, but we try and avoid storing it for any length of time as the free amine as a solid sample or in solution. So yeah, we're not sure actually. Haven't done a kind of having the NMR tube sitting around kind of analysis yet.
So from a procedural point of view, are we best making up solutions fresh and using them immediately, or can we
if
store
you
them
can.
in DMSO?
Well, so yeah, no, I think your DMSO reduced temperature out of the light, away from air, should be fine.
Yeah.
But if you can avoid making up all 6 milligrams, maybe, or whatever it was that was shipped, and maybe keep some as solid sample, that might help a little bit.
Okay.
Yeah.
Oh, may I have a question here? So about shipping these kind of like samples that are easy to deteriorate, is that possible? Maybe next time we'll just ship things like in the DMSO or something. If
It's always fine as long as you know what the concentration is. DMSO stock at high concentration is always fine, right?
Yes.
Yeah.
Okay, right. Thanks.
If you can get it to 100 millimolar, that's good.
I'll try.
Okay, so I think these are the 2 that went, so you've got those, which is great. So this is a control compound.
Yeah, they arrived this morning.
Okay, I mean this will be stable, no problem. It's just the amine that's the issue. So Yuhang is now the chemistry is starting to behave itself, and the purification is easier. He's planning on other diamines here. You know, in case this linker length is wrong, there are other diamines you can get with bot groups on one end, and we can make other compounds now. Yeah. All right, great. Thanks, Yuhang. And you know, it's great that you've got a sample of this guy. It has been much more difficult than we imagined getting that.
Yes, yes. Also, the picture of Yang's crystallography, I'm not sure whether that compound is. Yeah.
Okay, so just to switch to that, because that's the other thing that we got updates from. I think we saw these. Yeah, so we saw these had been done last time. This is structures from Yan at SSGCID, and I think last time reported that these were done, but they hadn't been deposited. So now they've been deposited, and they can be accessed using these URLs here, which is great. And the ligands that are involved in the binding to Mur-C were these 2 AZ compounds shown down here. Whoops, sorry, I skipped past it, didn't I? Oh, sorry. Where am I? I should be looking at the right. It's up here, right? Sorry, yeah. There. So these are the 2 Mur-C structures with these 2 compounds bound. In case anyone wants to double check. So obviously, you know, we're looking at structures where most of this unit is perfectly overlaid, and the side chain is flexible and out near solvent, right? And I guess we were interested in whether or not the side chain was picking up any other interactions, but ultimately they're bound in the way we might expect. As Joe mentioned, I think Yuhang got to specify these MIC values were against efflux efflux strains with the efflux pumps taken out, right? So not wild type. That's what these numbers mean, right? Just to clarify.
Yes, I think it's it is efflux deficient.
Yeah. Right. And so that's wonderful. And then the last email just came in today from Yan to say that there's another compound that he has a structure with Mur-C of, and gave a little screenshot here. Is showing this as the side chain. Right. So it looks like, well, it looks like there's a carbonate there and a ring with an alcohol pendant, with an amine pendant. So that's this guy. And so that has also now been co-crystallized along with these 2. They're all got the terbutyl group on, right? I thought we had a slight mix there, but you're sure about these structures, right? Because these are the AZ samples that were kindly donated, and they match to structures that were in the AZ Hamid paper.
Yeah, these 2 you're currently showing, I'm pretty sure, because I double checked with PDB.
Yeah.
And with the paper. So I found the structures exactly matching these two. The problem is, are we sticking to the AZ compound numbering, or are we sticking to the AZ commercial compound numbering? Because
well, it needs to be just cross-correlated, right? So we just need to make sure that both numbers are used so that we don't get confused. So these numbers are from the paper, but that's just the paper, right?
That these numbers are easy to track because when I search for the commercial, for example, the ligand AZ1364 or whatever, I cannot find any website resources that can just link to this structure. So I think maybe we stick to the paper numbering. It's a bit easier for us to identify.
Yeah, I mean, it doesn't really matter so long as we are clear about, we sort of cross-correlate the structures. That's all. Then it's fine. So I think that, yeah. So I think Joe sent us a file. Wait, just let me check here. One sec. Always, every single project, you have issues with correlating numbers between different coding systems. So Joe, kindly sent this earlier. And so this is the compound that we're talking about. So the compound 26 in the paper with that structure, which based on the picture that Jan sent looks right.
Yes,
yeah.
Yes.
I think that's it. So they're all got the same core and different side chains, which is great. So that is a very nice set of compounds with multiple compounds bound to the same enzyme, allowing us to really see what impact the side chain makes, if anything, but they're all bound the same way, I think. Okay, so if that is the, so if that's correct, then Yuhang, then we're all good based on the structure that you've put up here. And so there it is, which is great. Thank you, Jan, in your absence. That's really beautiful structure. So I think he's going to finish this off and then, submit it as per usual, so that we can then do the full overlay here. 1 second, sorry, I think there's someone trying to come in. That's right. Okay, great. All right, so I think that those are the main things. So data from Warwick, and then the new protein structures. Given how much we have had happening over the last few months crystallographically, and trying to move towards writing up this stuff in a way that is more than a sequence of PDB files and submissions, but putting it into something that is a sort of coherent manuscript. It would be nice to have another soak-in to talk about the crystallography. If everyone's okay with that, I can send around some times and get us to meet up. I think that was one of the sort of action points from the last time, was to try and make sure we met up again and talked about latest results. So I will do that unless I hear back. On the only other thing, I think we're going to mention, Daniel, you posted something about your synthesis of the competition compounds, and you needn't go through it in detail here, but the upshot is of these, where are we just with the final compounds? Because I guess we could still have these evaluated at a future date.
What I'm working now is the last step, actually. The reaction with MCPBA. I just saw in the morning that I have the epoxide, which is a good sign that the reaction is working. I'm keeping running the reaction. I add more MCPBA and TSPA, and I'm focused on to get at least one of the compounds, because the other synthesis I'm also have some problems. But I'll try to get this diazole first and keep working the other synthetic roads, because if you go a little bit down, this oxadiazole, I needed to change the first step because I was planning to do a reaction with acetic anhydride and then bromination with NBS, but didn't work. So I repeat the reaction with the 2-bromo acetic anhydride. It worked. However, purification is a bit confusing now, but I could see I have this compound LCMS, and what I'm thinking to do instead of synthesize again the butenamide, I'm gonna do or Gabriel seeds or whatever to convert the bromine to. I'm trying to use the azide to convert bromine to the amine, and then I can use the crotyl bromine, because the first step that I was working to synthesize the butenamide, the yield is so really bad, it's like 5%, and I'm trying to do this step first, and then I can go to the diol for the oxadiazole as well.
Okay,
and
great.
the last step also a couple of problems. The first synthesis didn't work, then I tried Suzuki coupling. Major product that I get was not the product from the Suzuki coupling. What I'm doing is now kind of protecting the bromine, the reaction worked, and then I'm gonna do the Suzuki coupling using this derivative. And the thiophene, I did the esterification of the carboxylic acid. Today I'm doing the bromination, and yeah, and then I will keep it going with this synthetic pathway as well.
All right. Yeah, I mean, these were the compounds that were predicted to be of high value according to Jan Jensen's work, right? So that's why we're still pursuing them, because they were quite nicely, they gave good scores. All right, great. Thanks, Daniel. All right, I think those were the main things here. Nothing else is yelling out at me as being needing to be discussed right now. But have I forgotten anything that needs to be discussed? Chris isn't here, so I can't pester him about some of these things in the middle here. Anybody want to mention anything else or ask anything which I haven't mentioned already? All fine. Just a note that as we get more of the more certainty on the inhibition by the enamine compounds from the Warwick enamine collection and the atomized compounds, you may have seen that Ed's already been doing some assessment of near neighbors of these. So, you know, I mean, what we really want to do, if we're happy with some of these hits, is to then start looking at, you know, what it makes most sense to buy. So he's already started looking at this for some of these compounds that were reported as being multi-targeting inhibitors, and just extracting the obvious compounds that we can buy that would flesh out some SAR. And obviously it makes sense to do that if we're really confident about the fact that they are binding and doing what we want. It's a judgment call about when we start with that. So on the left here are the ones in the Warwick collection, and on the right are the atomized compounds. It's a judgment call, right? Because each of these, it's relatively quick and easy to get enough material at about, you know, 100 euros per sample or something. And in the grant that we've got from Antibiotics Research UK, we have money to spend on this stuff. I mean, particularly on the atomized compounds, but we can use it for anything. So we do have money to purchase a bunch of compounds, and we are going to need to do that before the grant runs out in January. So keen to do that. And we just need to sort of make a decision about when we are confident enough that the compounds are really worth looking at, and then we buy the compounds around it. It feels to me like we're not quite there yet. Is that right, Laura and Adrian, that you'd like to do a little bit more analysis of the results, or are we there yet? We can start ordering a few compounds.
Yeah.
I mean, so we got the compounds that we're talking about. We have validated against the coupling system. The only other thing that you might want us to do, or we might do, is do some of the IC50 work and possibly some competition work to see which substrate binding site's being targeted. That said, you still have a group of molecules which do multi-target, and that part of me thinks that we should probably go ahead now, but that's so. I guess I'm in two minds. Sorry about that.
I guess if we want to have a competition results on them,
Yeah.
then more work will need to be done. But I guess the binding, well, the activity that we have now is validated.
Yeah,
right?
it is.
Right, on both. So on your initial, the Warwick ketamine collection and the atomized compounds.
Yeah.
Yes.
I mean, I think if it were just me, I think I probably will go ahead, to be honest. I think if simply because I know that they actually do work as inhibitors and they multi-target. The question about how they're doing it is something that we still have to flesh out, but nonetheless they do. And this is what we've in principle been after.
Yeah. All right, I'll dig up how much money we've got and how many compounds we can buy, and then we can buy a smattering, I guess. That will be good.
Yep.
I mean, I agree, because these came from larger collections where there were many compounds that didn't do anything. And so, there's plenty of negative controls, right, of compounds that were part of the same screening set didn't do anything interesting. So it's a good sign that we should investigate compounds more. And of course, if we do start playing around with these and we get some activity that is related, then it means we don't have a sort of suspicious looking singleton, and we might be able to get a little bit of SAR going.
Yeah, I think so.
All right, great. All right, Ed and I will look at that tomorrow. And we'll make some suggestions. Yeah, and we'll post what the plan is before we do it. All right. Wonderful. Okay, anything else, or are we done? Last call.
Yeah, we're okay.
All right, good. All right, in that case, thanks for coming along, everybody. See you next month, and I'll send out some possible dates for a focused crystallography discussion. Obviously, I'd like to get the SSG CID people along and Lesborough along and make sure everyone's involved in that. So I'll try and find a decent time for that.
Thank you.
Okay.
All right, thanks very much, particularly you guys, and more. Thanks very much for the data, really good.
No problem.
Bye.
See you.
Bye.
Thanks,
Bye.